Hello, everyone. Welcome back to Virology Live. Today we're going to talk about genomes and genetics. This is session number 3. Welcome, everybody. This starts today with something that happened in the 1950s, actually. 2 different experiments which showed that the viral nucleic acid genome is the genetic code. Now, many of you may know that DNA was discovered in the 1800s, but for many years people thought it wasn't the genetic material because it was too simple. It was made up of 4 different bases. People thought proteins were actually the genetic material. It wasn't until 1944, Avery McLeod and McCarty's experiment where they took DNA from a bacterium, introduced it into another bacterium, and showed it could transfer the property, a specific property via the DNA, that DNA was shown to be genetic material of bacteria, which of course was met with skepticism for many years. And then in the 1950s, 2 experiments were done to show that the viral nucleic acid genome is the genetic code. And there are 2 because, of course, we have both DNA and RNA viruses. So the first, the very famous Hershey Chase experiment with bacteriophage T2 and with tobacco mosaic virus. Remember, the first virus to be discovered, Frankel Conrad and his colleagues did an experiment to show that RNA was the genetic material. So let's explore those. We're going to go and dig into them a little bit. And yes, I did get a haircut yesterday. It's just easier to deal with. It's shorter, and it's easier to put headphones on. It's easier to put I have ear inserts here, which are way easier to get in. It's easier to dry after washing it. And yes, I kind of channel Pete Townsend. I really like that look. I think I've had the same haircut for ages. It's kind of crazy. All right, let's go into the experiments. Let's get rid of Vinny here. Sorry, we need number 3. This is the Hershey Chase experiment, a very famous experiment done by Alfred Hershey here and Martha Chase in 1952. And if you visit Cold Spring Harbor out on Long Island, the laboratory, they have a library, and in the library behind a glass, in a case behind glass, they have the apparatus that was used for this experiment. It's a blender. And there's a little letter here explaining all about it. It's a kitchen blender, which was key to doing this experiment. So here's how it worked. They want to know what's the genetic material of a bacteriophage. Is it the protein or is it the DNA? So they grow the virus, the bacteriophage T2, with either radioactive sulfur or radioactive phosphorus. And of course, when you buy radioactive sulfur in the form of amino acids that have isotopically labels on them, and you can measure that in various ways. So you can label the bacteriophage with sulfur, radioactive sulfur to label the protein, or with radioactive phosphorus, which of course will label the DNA. And then you infect E. coli with these bacteria. And the way this phage works, it attaches to the outside of the bacterium and injects its DNA inside. We know this now. Of course, they didn't know this at the time. But they said, okay, we're going to let the phage stick to the bacterium for 5 minutes, and then they put it in the kitchen blender, blending, and that shears off the bacteriophages, so you just synchronize the infection. And then you let the bacteria incubate, and the infection proceeds. The bacteria are lysed, the phages come out, and you say, well, where's the radioactivity? Is it in the phages? And when you label the protein, you find no radioactivity in the next generation of phages. However, when you label the DNA with radioactive phosphorus, you then find that there is radioactive DNA in the progeny phage. So this was interpreted to mean that DNA is the genetic material now. This, of course, was also met with skepticism, but eventually it held true. It was repeated, and we now know, of course, that DNA is the genetic material of DNA viruses. What about RNA viruses? A slightly different experiment, but same outcome. So tobacco mosaic virus is a rod-like virus consisting of RNA, and that's labeled here in green. We actually have conventions in this course for nucleic acids, which we'll see in a moment. The RNA, and then there's the protein coat, one protein that coats the RNA and forms the rod-like structure. And Franco Conrad had figured out that actually you can separate the protein from the RNA, purify each one, and then reassemble them. You can mix them back together. They will form an infectious virus. Okay, so he said, all right, let's do this experiment. He had two variants of tobacco mosaic virus. We'll call that the green variant and the red variant. And he could distinguish the proteins of both in a way that would tell him which variant it was. So he disassembled the green variant and the red variant, and he cross-mixed the protein with the genome. So he mixed the red protein with the green genome, and he mixed the green protein with the red genome. And he got infectious particles. So now you have green RNA with red protein, red RNA with green protein. He would infect tobacco leaves and ask, what comes out? Yeah, this is a misspelling. Sorry. It just spell check, right? Which has been plaguing me for years. Yeah, Franco, F-R-A-E-N-K-E-L. So let's do this in real time. F-R-A-E-N-K-E-L. And the spell check is the bane of correct spelling. Isn't that funny? All these words that it doesn't know. And I often want to turn it off, but don't know how to. Infect the cells with these hybrid viruses and see what comes out. And what Franco Conrad found was even if you put a red protein on the green genome, what came out from this infection was green genome with green protein. And what came out of the other infection was red genome with red protein. So the genome specifies the protein. Really nice experiment. And so now we know that both DNA and RNA are the genetic material of viruses. But what's going to guide us today, and in fact, perhaps even the bigger surprise than what we've just talked about, is that even though there's so many viruses on the planet, right? We have talked about all the different sorts, DNA and RNA, and we're going to talk more about that today. So it's almost infinite complexity, right? It turns out there's only a finite number of viral genomes. And because I teach my virology class here at Columbia University in New York City, I use the symbol for the number 7 subway line to remind students that there are 7 kinds of viral genome. What better way to remember that, right? And you guys who are all around the world, it doesn't help you so much, but I'm keeping the convention because I like it. And I have to say I take the subway frequently, and every time I see the number 7 train, man, I think about 7 viral genomes. And we're going to use another subway sign tomorrow, next week, Monday, when we talk about virus structure. 7 different kinds of viral genome. That's all there are as far as we know. And today it's going to guide our thinking. So there's 7 kinds of genome, and the reason this is important is all of them have to make mRNA that can be read by cellular ribosomes, right? Viruses are parasites of host cells. In fact, they're translational parasites. They have to be translated by cellular ribosomes. And there's our ribosome again, our turkey, which is a cellular product. And as far as we know, this applies for all viruses on the planet. Genomes have to make mRNA, whether it's a DNA genome or an RNA genome. we'll see in the next few lectures how you get from a DNA genome to mRNA, how you get from an RNA genome to mRNA. It's really interesting. We'll start to explore it today, actually. They have to make mRNA, and that's the key for this 7 different classes of genome. In the 1960s and 70s, David Baltimore, he's shown here on the upper left, he won the Nobel Prize in the 70s for co-discovering reverse transcriptase along with Howard Temin. We'll talk about that discovery later in this course. Full disclosure, I did a postdoc with David Baltimore from 1979 to 1982 at MIT. I think he's an amazing scientist. And he put together this, what we call the Baltimore Scheme, a simple way to think about virus genomes. He put mRNA in the middle because all virus genomes have to make mRNA. And then he put around it the various kinds of viral genome. He knew of 6 at the time. He didn't know of the 7th type, which was added later. So that gives us 7 different kinds of genome. And he could trace the pathway from the viral genome to mRNA quite easily. And there are 7, starting with 1 here, double-stranded DNA. We have single-stranded DNA, and then type 3 is double-stranded RNA. Type 4 is plus RNA that goes through a minus strand intermediate. Type 5 is negative-stranded RNA, and type 6 is plus-stranded RNA that's converted to a DNA and then to mRNA, and that, of course, is done by reverse transcriptase. So those are the 7 kinds of viral genome that we know about. We're going to go through each of these today. And it's useful for you to remember this picture. I have it in my head always, because then when you encounter a virus, you can immediately trace the pathway to mRNA. It will help you understand it better. Now, before we dive into the details, let's make some definitions again. We have to make sure we're talking about the same thing when we say certain words. First of all, we have these plus and minus conventions for RNA and DNA. What does that mean? Students often get confused. They say, oh, does it have to do with charge or what? No, it has nothing to do with charge. It's just a convention. And the convention was established many years ago that mRNA, which of course by definition is ribosome ready. mRNA is what is translated to protein on a ribosome using a process we'll talk about later. That's the plus strand. mRNA is always the plus strand. No exception, folks. Not changing it. Plus strand means simply mRNA ready. That's all it means. Nothing to do with electricity or anything else that you associate with plus. So DNA of the same polarity is the plus strand. And when you have a double-stranded DNA, there's a plus strand and a minus strand, which is the complement. The plus strand is the same polarity as the mRNA. So then the complements are called negative or minus strands. That doesn't matter which word you use. And so the plus strands complement. And by complement, you should understand for nucleic acid, the complementary strand is base paired with the first one, right? By the conventions of nucleic acids, which we'll get into a little bit here, but that's one thing I assume that you understand, that a double-stranded nucleic acid, whether it's DNA or RNA, is base paired by chemical interactions between the 4 bases that make up the DNA. So the minus strand is simply the complement. All right, so having told you that mRNA is the plus strand, I want to now tell you that not all plus RNA is mRNA. In other words, not all plus RNA is translated. And we will see how that works as we go through some of these viruses. All right. Now, the elegance of this Baltimore system, and I tell you, this is just a brilliant scheme. It has endured. It's not a classification scheme as such. It's not it's not a taxonomic classification scheme. It is a way to organize the genomes, and these go across families. So it's not about taxonomy. It's about mechanism. It's about understanding how these viruses reproduce. And the elegance is, if I tell you what the genome is of a virus, you can immediately trace the steps that we need to produce mRNA. Now, you can't do it yet because you need some more facts in order to do that. For example, the only DNA that can be made into mRNA, that's a process called transcription that we'll talk about later. The only DNA that can be made to mRNA is double-stranded DNA. You can't transcribe single-stranded DNA. You can't transcribe this hepatitis B virus DNA because it's gapped. It's not complete. Only double-stranded DNA can be transcribed to mRNA. Negative strand RNA, single strand, is not mRNA, so it would have to be copied to make mRNA. Now, plus strand RNA can be translated directly. And in fact, for these group 4 viruses, which include the coronaviruses, it is translated directly when it enters the cell. However, this group 6, the retroviruses that utilize reverse transcription, that RNA is plus stranded, but it's not translated. That's what I mean by not all plus RNA is translated. We'll get into that in a bit later. And finally, double-stranded RNA cannot be translated. Even though it has a plus strand in it, the ribosome can't get at it because it's bound up by the negative strand in the duplex. Please remember that. It's really important, and I know many of you will forget. I know this because on tests, students forget that you can't translate. You have to make a single-stranded mRNA from this duplex. So those are the rules that you need to know to see how any genome gets to mRNA. So that's our 7 classes of viral genome. We have our double-stranded DNA there. We have gapped double-stranded DNA. And the fact that it's gapped is not the only reason why it's a separate class. It turns out that this class of genomes, 7, also involves reverse transcriptase. So it's very different from all the others. We'll get into that later. And then we have single-stranded DNAs. If you listen to the last TWIV about, which was bespoke capsids delivered. You ever want a bespoke suit? You know, you go to London, you get a bespoke suit. Well, we can make you bespoke capsids made for you. Those are single-stranded DNA viruses, those adeno-associated virus, AAV. Then we have double-stranded RNA genomes, and we have single-stranded, 3 kinds, minus polarity, and then we have plus RNA either with or without reverse transcription in the reproduction cycle. So let's see what we have rescued from this little discussion so far. And let us see if Socrative is going to behave today. I understand the internet was messed up Monday, right? Or certainly Facebook was down. Maybe that was part of it. All right, question 1. Why is mRNA placed at the center of the Baltimore Scheme? A, because all virus particles contain mRNA. B, there's no specific reason. C, because all viral genomes are mRNAs. D, because mRNA must be made from all viral genomes. E, because Baltimore studied mRNA. So, again, the login for this is Socrative.com. You can get to it a couple of ways. You can do bsocrative.com/login/student, but if you just go to Socrative.com, it'll ask you for student login, and you put in a room number. That's all. Should be straightforward, folks. So answer that, and I will check out the chat here to see if we have some questions that will enhance our Oh, yes. I meant to tell you, 100,000 subscribers. Thank you, bro. You know, your name looks like Arnaut, too. Here we go. I wanted to show you this. 100,000 subscribers on YouTube. This happened Monday, just after we finished the class. How about that? And I am just really impressed that so many people want to learn about science. You know, at the beginning of 2020, I had just over 20,000 subscribers. So it was really the COVID bump, sadly, that pushed it over 100,000. And now I think we have a nice group going forward, and I'm sure we can get to 200,000 and more. So what is the cell pellet? Yes, I think there is a slide there where I think it's, in fact, the let's go back to it. I have the ability to go back to any slide that we want. I think, yes, radioactivity predominantly in the cell pellet. So you take your infected cells and you centrifuge them. You spin them in a tube so that the cells will go to the bottom of the tube, and then you can remove the liquid from the top, which we call the supernatant, and the pellet are the cells that you've separated from the liquid. That's what the cell pellet is. It's a good question. It's something that I think a lot of people would get confused by. All right, in the Baltimore classification, do most of the families in each class group closely on the phylogenetic tree? No, they don't, actually. That's what I meant by saying this is not a taxonomic classification. It is simply a way to classify genomes. In fact, there was a very nice review article by Eugene Koonin not too long ago where he talked about the Baltimore Scheme. Actually, I reviewed it, and I thought it was wonderful. And he says, it doesn't tell you about phylogeny, but it's a good way to keep the genomes in order. The phage R2 system by Benzer. Man, let me tell you, when I was a student, an undergrad, I loved those papers by Benzer. No, I'm not going to, but not in this course. But we can do it in a future course. We can certainly do it in the next course, which may be on specific viruses. And I'm going to teach more courses because this seems to be working. There are people here who seem to be interested, although the number of people here at every lecture is decreasing. But I suspect we will get to about 150, and that's fine. I don't need to teach billions of people. Can you describe double-stranded RNA and single-stranded DNA? DNA is always double-stranded, RNA is always single. No, it's not true. It's not correct. Actually, I'm not sure you can see these questions on the white background so well. I'm sorry if you can't. There is single-stranded DNA and there is single-stranded RNA and double-stranded RNA, and the viruses are the ones that showed us about that. So it's just not correct. And I think I've given you some information on that. All right. So how does lacrosse virus work? We'll cover that. Maybe today. I'm not sure, but we're going to cover it absolutely. No minus. Yeah, well, in fact, single-stranded DNA viruses package both strands, one or the other. So you can't really say there's a plus and a minus. There's one or the other. Yeah. Does the plus or minus indicate the 5 or 3' end? No, it has nothing to do with the ends. It's just the polarity with respect to mRNA, right? Which of the 7 occur in plants? Well, we will get to that actually today. I'm going to show you a picture of which of the classes appear in different kinds of organisms, hosts. All right. Let's get to our quiz now, which should be done. We have 83 out of 99 students. That's good. Let's see what we got. So most of you got the right answer, which is D. Why is mRNA at the center? Because mRNA must be made from all viral genomes. It's not because all virus particles contain mRNA. 12% of you said yes. If you look at the Baltimore Scheme, those nucleic acids are what are in the particle. Actually, I didn't say that. So let me say it now. In the Baltimore Scheme, the 7 genomes are what's in the particle. And so mRNA is not in all of them, right? Some of them have DNA and some of them have single and double strand, the single and double strand RNA, and so forth. There's no specific reason, not at all, because all genomes are mRNAs. No. because Baltimore studied mRNA. Well, he did, but that's not why he put mRNA at the middle. That would be terribly self-serving, right? And we know that scientists are driven by ego for sure. Baltimore, in fact, said that himself. But that's not why he put it there. I'm looking at this comment here. 100K plaque assay would be awesome. What do you mean by 100K plaque assay? Do you want me to do one live? That would be fun, but it would take some time to get going. Anyway. All right, that's our first question. Let's get back to the lectures or the session. I don't want to call these lectures because it's unique. Viral DNA or RNA genomes, you can have both. They're structurally diverse. Besides being in the 7 classes, they can be linear. So we can have linear DNA. We can have linear RNA. They can be circular. We can have circular DNA. We can have circular RNA. They can be segmented, which means they're in pieces in the particle, the virus particle. They're in pieces, as opposed to one molecule in many virus particles. They can be gapped, as for the hepatitis B viruses, where one strand is not fully double-stranded, and you're probably thinking, what does that mean? We'll get to it. We'll get to most of these things. They can be single plus or minus, of course. They can be ambisense, and this is the Bunya viruses that someone talked about. Ambisense means it's an RNA with part of it is plus-stranded and part of it is minus. What does that mean? The plus strand means it's the same polarity as mRNA. The minus strand is the opposite. And why would you do that? What would be the function? Why we don't ask why questions in biology. You can ask why in philosophy, but this is not philosophy. You ask, what's the function of having that? And I don't know. I don't think anyone knows. It works, though. In many of these genomes, you'll see, you may ask, what's the function of having this diversity? It works, and it's selected in different niches, most likely. Double-stranded genomes, we can have genomes with covalently attached proteins. We can have genomes where the ends are covalently linked, chemically covalently linked. So this is a poxvirus double-stranded DNA where the ends are bound to each other, covalently joined. So if you separated these two strands, you would have a single-stranded circle. And another one here, DNA with covalently attached RNA, the hepatitis B genome, in addition to being partly single-stranded, it's got a piece of RNA on it, it's got a protein stuck on it. That is, to use one of Amy's words, that seems to be fracacta, right? But it's not. Nothing in virology is that. It all makes sense when you look at how it works. So we'll talk about that later. Now, here's a question that you probably want to know. You want to know what's the function of genome diversity. Notice I didn't say why. I want to know what the function is. Well, what's the function of having both DNA and RNA? I can tell you that because RNA genomes were the first on Earth before DNA arose. There's no doubt about that. There was an RNA world first. There was an acellular RNA world where there were just molecules reproducing in the soup that was ancient Earth. RNA appeared first. It was a whole world based on RNA. We don't know how complicated it was, but at some point DNA arose, and that most likely arose through the evolution of the enzyme reverse transcriptase, which could convert RNA to DNA. So that meant that in the RNA world there had to be proteins eventually. Whether or not there were RNA cells, we don't know. Cells require big genomes. There could have been. But anyway, the key is that RNA came first, then DNA, and RNA viruses are relics of the RNA world because they're still around. And obviously they're evolutionarily competitive. That's why they are still here. We have even more direct evidence of an RNA world in the entities called viroids, which we will talk about later in this course in some detail. Viroids are relics of the RNA world. They're just RNA, and they don't code for any protein. Here's one. There's no capsid. So that's why I say a viroid is not a virus. Eugene Koonin actually convinced me that not to look at viroids as viruses because they don't have a capsid. He thinks that's one of the defining features of a virus. So the viroids are naked RNA, code no protein. They get into plants mainly, and they reproduce. So that's probably a relic of the RNA world. Now, if you may ask, what's the function of having linear, circular, segmented, double-stranded, single-stranded, plus polarity, minus polarity genomes? I cannot tell you the specific functions. I don't think anyone knows. What only we can say is they all work, they all exist, they're evolutionarily competitive. If they weren't, they wouldn't be around. Now, if you asked me to design the perfect virus, which nobody can do, by the way, I'm going to do a thought experiment. If you asked me to do it, I would make it a plus strand RNA virus because as soon as the plus strand gets into a cell, it can be translated and the genome reproduction cycle, the virus reproduction cycle can begin. All the other viruses have an intermediary step, which is more or less complicated. Let's take a negative strand RNA virus. The negative RNA gets in a cell. It can't be translated. It has to be copied to plus RNA. It's more complicated. DNA viruses, many of them have to go into the nucleus, and then mRNA is made. So to me, nothing makes sense except a plus strand RNA virus, which is another reason not to anthropomorphize. Do not put human values on viruses because you could be wrong. See, I said plus strand rules, and they don't. Well, they do rule in the sense that they cause most of the disease on the planet. So that's the story here. I'm going to talk more about viroids. People are asking about viroids. There's a whole lecture on viroids later in this course. So I think you ought to memorize these 7 families. But nowadays, nobody has to memorize anything. You could carry it around with you. But realistically, I know none of you will want to do that. However, I always tell my medical students when I teach them, I said, what if someday a patient asks you, what does it mean that this virus I have is double-stranded RNA? And you don't know. You're going to look silly, right? Now, the chances of that happening, who knows? But a student may want to know, and you should know it. It's one of the things you should know. Plus, it helps you understand viruses in general. You can be asked about any virus, and you can start to talk about how they reproduce. If you know the genome structure, you should be able to figure out how mRNA is made and also how the genome is copied. In fact, in this picture, not only do we have the pathway to mRNA, but we can tell you how the genome is reproduced. So, for example, for a single-stranded DNA virus, you have to go through double-stranded intermediates. For a gapped, you have to become repaired. For a plus-stranded, say a poliovirus, you have to go through a negative-stranded RNA. Minus RNA has to be copied. Plus double-stranded RNA has to be copied to an mRNA. So you get information about reproduction as well. Linus Pauling used to say he never memorized something he could look up in a book. I think in general that's fine, but there are some things like this. Well, first of all, it's easy. This is a piece of cake. You don't have to memorize anything. You just think of the 7 genomes. You could think of them at the top of your head, and it helps you to figure out things on the fly. I think if you're teaching, if you're in discussions, if you're on panels, you can't look something up in a book. You need to have it in your head. And we do a lot of that nowadays, right? So some things you need to know. I'm sorry, Linus. Maybe Linus lived in a day where he didn't talk to anybody live, you know. You give a talk, you have your slides, you have everything you need. All right. What information is encoded in a viral genome? And I don't care that he won 2 Nobel Prizes, by the way. Winning a Nobel Prize doesn't mean you know everything. We know that because recently a Nobel laureate got something very, very wrong. In fact, several Nobel laureates have recently got things wrong about SARS-CoV-2. Okay, point taken. What information is encoded? Well, some of the things would be things that you need for protein synthesis. This is mainly giant viruses. They code for parts of ribosomes. In fact, one virus, as we mentioned last time, encodes all of the proteins you need for translation in parts of the ribosome. You have to encode proteins to reproduce the viral genome because cells cannot do this completely for the most part. viruses always have at least one protein that is involved in replication of the genome, packaging of the genome into a particle, assembling the particle, timing and regulating the replication cycle, modulating host defenses. Every virus has to encode at least one modulator of host defenses, otherwise they're gone, because host defenses are so good. It's really, really true. We'll get to that later. And proteins that are involved in spread to other cells and hosts. So basically, proteins involved in the whole reproduction cycle have to be encoded in the genome. But you don't have to have all of these. All right, don't look at this list and say, do all viruses have to do this? The adeno-associated viruses encode two proteins. They have a protein involved in DNA replication, the rep, and they have a capsid protein. They don't do any of this. But some of those proteins may be multifunctional. It's likely that they are in terms of modulation of host defenses. Yeah, actually, Tupan virus we haven't talked about yet. Only the ribosome is lacking. This virus, a giant virus, it's beautiful, right? How beautiful is this? Well, of course, it's colorized by the people who took this electron micrograph. It's got a spherical capsid and some kind of a tail. It is not analogous to the bacteriophage tail, by the way. This genome, big genome, encodes 20 amino-acyl tRNA synthetases. These are the enzymes that link amino acids to tRNAs, right? The tRNAs are the adapters that read the genetic code on the mRNA. 70 tRNAs are encoded in the genome. Multiple translation, initiation, and elongation proteins. If you don't know what that means, these are proteins essential for translation. The cell encodes all of these, of course. Genes that encode factors related to tRNA and mRNA maturation. This is the most complete translational apparatus of the virosphere. This was discovered by a French and a South American group together, and the French have a lovely way of naming things and saying things, right? As I've already said, Mimi virus, Mama virus, Moo Moo virus. It's really cute. And here, this phrase, only the ribosome is lacking, it's not how we would say it in English, right? It's a direct translation of the French. But I think it's cute in a way, quaint maybe. Only the ribosome is lacking in the genome of this Tupan virus. Now, you may ask, why is that? Is it still a virus? Of course it's still a virus. It depends on a cell. If it didn't depend on the cell, it wouldn't be a virus anymore. Then it would be a cell. Why does it encode? Or what's the function? See, even I say why now and then. What's the function of encoding all of these translation-related items? Well, probably to tweak the cellular translation apparatus so it favors the virus. We don't know because no one has yet done the right experiments to address this. Well, what's not contained in virus genomes? No genes encoding the complete protein synthesis machinery. I have to tell you that years ago when I started teaching this course, on this slide I had no genes encoding any of the protein synthesis machinery. And that's because we hadn't yet discovered giant viruses. So we didn't know that some viruses could encode it. Everything is fluid. So, but no virus encodes the complete protein synthesis machinery. There are no genes encoding proteins involved in membrane biosynthesis. There are no centromeres or telomeres that we find in our host chromosomes. The centromeres, these structures in chromosomes, they're not in viral genomes. I used to have on this slide no genes that encode metabolic components. That's gone out the window. Lots of giant viruses encode parts of the Krebs cycle. We talked about that last week on TWIV. However, caveat here, we probably haven't found any of these yet. When you sequence the genome of a giant virus, 90% of the genes don't look like anything that you can recognize as being functional, as having some function. And so it may be that we haven't discovered some of these items, like genes involved in membrane biosynthesis. Maybe they're very different from the enzymes that are in cells. So stay tuned. It is a rapidly, constantly changing field. Now, last time someone asked, what's the biggest genome? What's the smallest genome? So I've had this slide in the lecture for quite a while, so we will use it here in our sessions. These are the largest known viral genomes. They're all DNA. The DNA genomes are the biggest. They outsize RNA genomes by far. So the biggest viral genome that we know of is from Pandoravirus salinus, which is a giant virus, and it has almost 2.5 million base pairs of double-stranded DNA. This is huge, encoding 2,500 proteins. I mean, compare that to adeno-associated virus, which encodes 2 proteins, small circular DNA. We'll see that on the next slide, I think. And then we go down from there. Pandoravirus dulcis, 1.9 million, to Panvirus, which we just talked about. Now, in between here is the genome of Haemophilus, which is a bacterium. 1.8 million base pairs is the DNA genome of this bacteria. Haemophilus influenzae, for example, smaller than these 2 giant viruses. Isn't that amazing? Now, it's a free-living bacterium. Then many other giant viruses, and you can see the cute names: megavirus, mamavirus, mimi virus, mumu, and that's all the French naming. And I'm not making fun of them. I think it's just great that they come up with these cute names. Not everything they do is great. Not everyone does is great, but this is cool. And then Cafeteria Roan-Bergensis virus, a picture of which I showed you on the first session. All right, those are the largest known viral genomes. Can we get bigger? Probably, yeah. It's just a matter of finding them. Largest RNA virus genomes, kind of pathetic. The biggest one is only 41,100 bases in length. 41,000. These are all, by the way, coronaviruses. It's not actually the right classification, but you all recognize coronaviruses. There's a higher classification that encompasses these, but these are actually not coronaviruses. They're called nidoviruses, but that's how coronaviruses are also nidoviruses. So the biggest is a virus from planaria. Do you remember planaria from school? Those worms, flatworms, where you can cut them and they grow back. Anyway, they have a virus. Everything has a virus. No question. There's a virus of aplysia, which is the mollusk that Eric Kandel and others have used to study memory. And there's a nidovirus of snakes, the ball python nidovirus. So these are the biggest RNA virus genomes. And so it doesn't compare to 2.5 million for DNA, right? Why? Because RNA is unstable. It breaks. It's physically easy to break, much easier than DNA. And that's probably why cells have DNA genomes, because the genomes have to be big, and an RNA genome could just not sustain a cell, probably not an organism either, a multicellular organism. What about the smallest viral genomes? Here they are. Well, so here we have viroid, which is not a virus, and I've had this on the slide just to show you how small they are, and they don't code for any protein. So these are relics of the RNA world. And someone asked, how do we know there was an RNA world? Well, this is one piece of evidence that these don't code for any proteins. And we know also that RNAs can be made in the test tube that can self-replicate. And then we're moving into viruses. We have satellite viruses. Now, a satellite was the original name given to a virus which depends on another virus to reproduce. It's lacking a number of functions, so we call it a satellite. So it has to be in the same cell infected with the helper virus. So this has been known for years. Viruses do not have their own viruses. They have helper situations where the helper virus helps another virus. They have to simply be in the same cell. So this satellite RNA doesn't encode any proteins, small genome, but it's encapsulated by proteins provided by the helper. So that's a virus. Then we have the hepatitis delta virus, which is a human pathogen, which we will talk about. It's an RNA of 1,700 bases, single-stranded, that encodes one protein. That's incredible how small it is. And then we get bigger. Here's a serovirus, which is a DNA virus, single-stranded DNA. We all have these viruses in us, seroviruses and anelloviruses. Your blood is full of them. They encode very few proteins. Then we have Gemini viruses, which are plant DNA viruses. Hepatitis B virus, leviviruses of bacteria. All these are RNA viruses, all quite small. So those are the smallest known genomes. It's time for another quiz. Socrative is working today. Very good. What information may be encoded in a viral genome? A, gene products that catalyze membrane biosynthesis. B, complete protein synthesis systems. C, centromeres or telomeres. D, enzymes to replicate the viral genome. So what may be encoded? And let's go to some of our questions. Let me see if I could find some questions where I left off last time. It's one of the issues that the chat scrolls here. So Tupan virus, I think that's correct, Sun King. It's a local word. I think from South America, the country where it was isolated. Yeah, I believe it means Sun King, something like that. You can look it up for sure. Do plus-strand RNA viruses have a greater mutation rate than plus-strand? Do non-plus-strand have a greater mutation? No. The mutation rate is dependent on the polymerase, not the genome. And we'll actually look at that. Virosphere is a good word. Yes. Virosphere, all the viruses, right? I like it as well. Who's the host of Tupan virus? Unknown. It will grow in amoeba, but it's not clear to me that that is the natural host. When this session is complete, could we talk you into doing a 201 series of gradually increasing difficulty of concepts? I would like to teach a virus course next, and perhaps that would have increasing difficulty as well. Yeah. Can a viral infected cell inhibit presenting receptors? Yes, but we're going to talk about that in the immune response for sure. Smallest viral genome I showed you on the slide, right? It's that satellite virus. Our DNA viruses larger because they're somewhat more durable, stable. Yes, DNA is chemically more stable than RNA, and so it can get longer and encode more proteins, and therefore you can get cells in complicated organisms. That is that's the idea. I like the comment about finding possible membrane biosynthesis. I'm sure they're out there. I mean, look, we have all kinds of metabolic enzymes. This would be there as well. And you can imagine that the reproduction would require a slightly different membrane, right? Does a larger genome imply higher evolution, increased functionality? I don't think so. I think all of these viruses can infect hosts, reproduce, and in some cases cause disease, although I don't think that's a sign of higher evolution. I think that's an accident. I just think these... they work for some reason. We don't know what the extra genes do, right? So if we did, I'm sure they have a function. So, but I don't think that necessarily bigger is better. I think they can all do they can all persist. They can all reproduce, and that is the key. What disease does Mimi virus cause? It's not a human virus. So it's a we don't know the host, but it reproduces in amoeba. There's some evidence that these giant viruses do reproduce in humans, but whether they cause disease or not is clearly not known. It was said that SARS-CoV-2 cannot mutate as much as flu because the genome is not big or varied. No, that's not correct. What was said, and we will come back to this, the spike, the hemagglutinin of influenza, is more plastic than that of the spike of coronaviruses. And that is very different from mutation of the genome, right? Mutation rate is different. But we will talk to that, okay? There are not viruses that attack other viruses. There are viruses that require a virus-infected cell in order to reproduce, and they may interfere with the reproduction of the helper. But I wouldn't say they attack them, and they don't reproduce in the virus. They reproduce in the virus-infected cells. Have any mitoviruses been found outside of fungi? I believe there have been in other hosts. They originated from, I'm not sure exactly what, but I believe so. All right, we have to... yeah, some people are answering the quiz questions. Oh, well. What's the difference between a viroid and a satellite? A viroid is not capsidated. It doesn't have a protein shell on it. A satellite does. So a satellite is a virus. Is it possible to add a numerical label? I was thinking of this. I label my lectures, right? But I thought of these as something where you could go to any of them and just watch it. So I didn't want to label them. Get away from what I have been doing in the class. I think it can be followed without a number. All right, so this last one. Are there viruses of mitochondria or chloroplasts? Yes, there are. Mitovirus is one of them, for example. Right, what do we have here? Show results. Not everybody answered. You should really answer, folks, unless you're not here. It's for your own good. Most of you got D, enzymes to replicate the viral genome. That is what may be encoded. Gene products that catalyze membrane biosynthesis, I told you, they're not encoded, not that we know of. Complete protein synthesis systems are not encoded. Neither are centromeres or telomeres. These are exactly what were in the slides. Okay, let's return to slides. Let's talk about DNA genomes. Double-stranded DNA viruses dominate the bacterial virosphere. Interesting, isn't it? So most of the viruses of bacteria are double-stranded DNA viruses. Now, of course, as they have double-stranded DNA, I write here, they emulate the host, meaning they reproduce their DNA much as the host does, and we'll talk about that later. But the DNA of viruses is not like a cell chromosome. Your cellular chromosomes are the DNA is wrapped around nucleosomes, which are made of histone proteins, and we call this chromatinized. I'm going to use that word a bit. It's kind of jargon, but it means the DNA is wrapped around histones, and that regulates the activity of the DNA, as we will see. But viral DNA genomes, for the most part, are not, with the exception of the polyomaviruses. And viruses do things slightly differently from cells in terms of... I'm sorry, I left the question up. My apologies. In terms of DNA reproduction, as we will see when we talk about that in these sessions. So here are some examples of double-stranded DNA genomes. So the part of the Baltimore Scheme that's relevant here, we have mRNA in green. By the way, so our convention will be plus RNA is this Kelly green, and the minus will be a sort of an olive green, as you'll see in a moment. The DNA, the two strands are plus or minus. The plus strand is the dark blue, the minus strand is the light blue. And then for proteins, we try to stay away from greens and blues. We use browns. This is all from the textbook, by the way. We work very closely with the artists to make sure it's clear. We can divide... before I go into that, the DNA is transcribed to make mRNA, which is then made into protein, translated into protein by the host cell. And then the DNA has to reproduce, replicate, so that it can be incorporated into new virus particles. We have two general types of DNA, double-stranded DNA genome that we recognize. We have on the left viral DNA genomes that are copied by the host DNA polymerase. These virus genomes are too small to encode their own DNA polymerase, so they are copied by the host. And here's an example. Polyomavirus genome, 5,000 bases of double-stranded DNA. It's too small to encode its own DNA replication system. But then there are double-stranded DNA viruses, of course, with larger DNA genomes, and they do encode their own DNA polymerase. These include the adenoviruses with double-stranded DNA genomes of between 30 and 50,000 base pairs. The herpes viruses, 120,000 to 240,000 base pairs, and the pox viruses, which are even bigger, 130,000 to 375,000 base pairs of DNA. Now, the giant viruses are even bigger, of course, and they do encode their own DNA polymerase as well. So 2 categories based on whether or not the genome encodes its own DNA polymerase. And DNA polymerase, of course, is the enzyme that makes DNA from DNA. Here are some double-stranded DNA virus genomes. This is adenovirus here at the top, which all of you now know of because adenovirus vectors are used for some COVID vaccines. All these have double-stranded DNA. These can cause a variety of human and animal infections. Herpes viruses, somewhat larger. Envelope viruses compared to the adenovirus, which is a non-envelope particle. Papillomaviruses, the agents of warts and cervical and anogenital cancers in humans. We'll talk about those later. Small double-stranded DNA genomes. Polyomaviruses with small circular double-stranded DNA genomes, which infect most of us. And the pox virus is much bigger. The pox includes smallpox virus, of course, which has been eradicated. And here's a giant virus. This is a pithovirus, which is actually not drawn to scale. It would be much bigger than this compared to the other virus. It wouldn't fit on the slide. I thought it was better to fit it. It's really, really much bigger than all the others. All right, those were double-stranded. Then we have viruses with gapped double-stranded DNA genomes, which include, these are the hepadnaviruses. Hepatitis B virus, a very medically relevant pathogen, which we will talk about more. In the virus particle, the viral DNA is partially double-stranded. It's circular. One strand is single strand. You can see it's the negative strand by the color. It has a piece of RNA on it. It's got a protein linked to it. And this DNA cannot be transcribed to mRNA. It has to be repaired first to become double-stranded DNA. The protein's taken off, the RNA's taken off, the gap is filled in. Then it can be transcribed to mRNA, which will, of course, encode proteins to build new virus particles. This virus has reverse transcriptase in its reproduction cycle. And so some of those mRNAs are actually copied to DNA by reverse transcriptase. And the DNA, the gapped DNA, is what's found in the virus particle. So this is a unique family. This is family 7 in the Baltimore scheme. And finally, the last of the DNA genomes, single-stranded DNA genomes. As I said, these can package either minus or plus strand DNA. It doesn't really matter because either can be made double-stranded, right? When you think about it, these DNAs are actually copied by host cell DNA synthesis systems. So whether you put in a minus or plus DNA doesn't matter because they're going to be made double-stranded in the end. And then, of course, the double-stranded DNA is what can be transcribed to mRNA to make some proteins. The double-stranded DNA will reproduce to make plus and minus single strands, which are then put in the virus particle. And these single-stranded genomes can be 2 configurations. They can be circular or linear. So here's a virus with a circular single-stranded DNA genome, quite small, 1.8 to 2.3 kilobases, 1,800 to 2,300 kilobases. And an example of a circovirus is called TT, torquetanovirus, which is a virus in every one of us. It's in your blood. Doesn't make you sick. What it's doing there, no idea. but all of us have it. Parvoviruses, single-stranded DNA with unusual ends shown here. We'll talk about these more. These include the B19 parvovirus is responsible for 5th diseases. There are 5 childhood rash diseases, measles, mumps, rubella, chickenpox, and then we have 5th disease. There's also a 6th one as well. It's a herpes virus. This is a single-stranded DNA virus, and also adeno-associated viruses, highly used as vectors for gene therapy, are also parvoviruses. All right, time for another question. Which DNA genome on entry into the cell can be immediately copied into mRNA? This is testing your knowledge of the Baltimore Scheme. Double-stranded DNA, gap double-stranded DNA, circular single-stranded DNA, linear single-stranded DNA, or all of the above. Which one can be immediately copied into mRNA? All right, and while you are doing that, we'll switch to the Vinny view and look at some questions here. By the way, I just want to remind everyone that you should go to the course website. Now, let's bring it up here because I think a lot of people are not probably looking at it. Let's bring up here. Here's the website, virology.ws/virology-live-2021. It does link in the YouTube, okay? This is the schedule. Each session, we got the quizzes. Every week there's going to be a quiz. Tomorrow, a quiz for these 2 lectures are going to go up, which you can take. It's a Survey Monkey thing. And we have the slides as PDFs. We have study questions. We have a link to the video, okay? And then these are readings that are suggested that will enhance your understanding of viruses. And counting viruses, that is my video on how to do a plaque assay, in fact, with Amy doing a plaque assay. Let's find Amy. There's her hands doing a plaque assay. You should go check this out. This is very cool. All right, so this schedule is good for you to look at because it has links to things that I talk about. Okay, so let's get out of there. Let's go back to Vinny and answer some questions. All right, can any giant virus infect humans? Not that we know of. We've isolated them from people. We don't know if they're actually reproducing. We don't know if they cause disease. Aren't virophages basically viruses of viruses? No, they are not. I've said this 4 times now. They are not viruses of other viruses. They are helper-dependent viruses. It's a different thing, and I want you to understand the concept because it's not the same as saying a virus infects another virus. A virophage depends on a giant virus to reproduce. It can't reproduce by itself in a cell. So it's helper dependent. And in the cell, the helper virus provides functions, proteins that are needed by the satellite or by the virophage. All right. And as I've said, some of these helper dependent viruses can inhibit the helper, which seems like biting the hand that feeds you, right? Doesn't make a lot of sense. But it's not. It's part of the beauty of ecology and how everything's are related to each other. So don't say that viruses infect other viruses. Don't say they're viruses of another virus. They are helper-dependent viruses. That's the way to look at it. So to say that viruses have viruses is not true. They don't have viruses. There's a helper-satellite relationship, if you will. And so, yes, hepatitis delta is helper-dependent. It requires hepatitis B virus. It can only infect a cell that is infected by hepatitis B virus. Right? Delta does not infect hepatitis B. It infects hepatitis B virus-infected cells. Now you may think, oh, come on, Racaniello, it's splitting hairs. No, it's not splitting hairs. It's a very different idea. You don't get Hep D without B. So let's see what else we have here. Are phages included? They don't make the cut. They don't make the cut, unfortunately. They do. They don't make the biggest or the smallest. Can I talk more about these viruses we all have? I'm not going to talk about them in this course because we don't know anything. It would be a good topic for the virus-specific course, which will happen later. However, we will talk a little bit about the viruses we have in us when we talk about persistent infections, for sure. Let's see. What's the difference between a viroid and a satellite? A viroid is just a piece of RNA that encodes no protein. It doesn't have a capsid. It doesn't have a shell. The satellite has a shell provided by the helper. Viruses do recombine. That's sort of like having a virus in a virus. No, absolutely not. It's simply a genome that's made from bits from different virus genomes. There's nothing, not even remotely close to being a virus in a virus. Chromatin is so cool. Is it recommended not to swallow phlegm? You can. That's part of your defense, actually, to swallow phlegm, because it comes up from your lungs with viruses in it, and then you swallow it and digest them. So go ahead, swallow your phlegm. All right. So again, can a virus hijack another viral genome? I'm not sure that hijack is the right word. The helper-dependent virus is dependent on functions provided by the helper. So adenovirus provides proteins needed by adeno-associated virus, and they have to be in the same cell in order to do that. So the gap in the gap DNA are missing bases on one of the strands. Exactly right. One strand is just single-stranded. It's missing the other strand. And why that happens, we'll find out later. How is it regulated which strand goes to the ribosome and which strand goes to produce new particles? This is a great question. So early in infection, you need to make a lot of proteins. So the plus mRNA strands are mostly being translated. And then for plus strand RNA viruses, where the mRNA is the same as the genome, like polioviruses, you have to divert them to being encapsulated later on. And we think that's a function of making a lot of capsid proteins which pull the mRNA away from the ribosomes. Very good question. Is it true that operations on DNA as opposed to RNA can only happen in the nucleus for eukaryotes? I don't know what you mean by operations. DNA is reproduced in the nucleus. It's transcribed to mRNA, to ribosomal RNA, to tRNA in the nucleus, and those are all exported into the cytoplasm. There can be post-transcriptional modifications in the cytoplasm, though, like degradation. Mucus is a defense. Mucus is cool, right? We had a podcast about that. All right, let's get to the quiz now. And for that, I need to go back to quiz and turn off the question. Okay, and now I'm on the wrong page. I'm on my streaming health page. Did you know that a stream can have health? Can be healthy or not? Which DNA genome on entry into the cell can be immediately copied into mRNA? 79% of you got the right answer. Double-stranded DNA. It's the only one of the DNAs that can be immediately copied into mRNA. Gap double-stranded, no, it doesn't work. The enzyme can't handle the gap genome. Circular single-stranded, good. Nobody picked that. Linear single-stranded. No, single-stranded doesn't work. Has to be double-stranded. Not all of the above. Okay? These quizzes are working today. It's good. Good news. Let's continue. RNA genomes. Of course, these are my favorite because I work on RNA viruses, but DNA viruses are good too. So we have this interesting factoid. RNA viruses dominate the eukaryotic virosphere, but they are rare in bacteria. Remember I said double-stranded DNA viruses dominate the bacterial virosphere. And in fact, RNA viruses are rather rare in bacteria. They exist, but they're rare. But there are a lot of RNA viruses in the eukaryotes. Why that is, not clear. People speculate on it, and maybe We'll talk about it later. So here are the different kinds of RNA genomes that we can have, single molecules, segmented, double-stranded, circular. And this is a cool figure that gives you kind of a distribution of all the kinds of viruses in different hosts: plants, invertebrates, bacteria, archaea, other. Other would be algae, fungi, yeast, and protozoa. They get to be others, unfortunately. Sad for them. So plus-stranded RNA viruses, 22 families in plants, 15 in invertebrates. See, not only one family in bacteria, none in archaea, 10 in the others. That's all right. Double-stranded RNA viruses, a bunch in plants. So someone asked before, what viruses infect plants? Certainly DNA viruses, but now you're seeing the double-stranded RNA. 4 families in plants, 2 in invertebrates, only 1 in bacteria, 10 in other. A lot of double-stranded RNA viruses in fungi. And then finally, minus-strand RNA viruses are in plants, invertebrates, none in bacteria, none in archaea, and only 1 in the other category. So there are differences in what kinds of viruses, based on their genome, infect different kinds of hosts. And we will somewhat get into that when we talk about evolution. It sort of explains that in some way. Now, a few key facts here that, this second bullet point is incredibly important for you to remember. Cells do not have RNA-dependent RNA polymerase, which I will abbreviate as RDRP from time to time. They do not have the ability to copy large RNAs into other large RNAs. So that has to be done by a virus enzyme, the RDRP, encoded by the virus genome. So every RNA virus has to encode an RNA polymerase, except for the hepatitis delta satellite virus, which is an RNA virus. It doesn't encode its own. It's actually copied by host RNA polymerase II in a weird twist. RNA polymerase II normally makes mRNA from DNA, so it's doing something strange with the delta virus, but we'll get back to that later. So viral genomes, RNA genomes encode the polymerase, and that's a great drug target because cells don't have it, right? And there are a bunch of antivirals directed against RDRP. And that RDRP makes mRNA and it makes more genomes. It does both. RNA-dependent RNA polymerase. So let's go through the different kinds of RNA genomes. We have double-stranded RNA genomes. Remember, double-stranded RNA cannot be engaged by ribosomes. The double strand makes the plus strand inaccessible, so the virus has to make mRNA from the double strand, which means it has to carry an RNA polymerase in the virus particle. Okay? Very important. This virus, here's a rotavirus, cause of human gastroenteritis. This double-stranded RNA virus has to have an RDRP in the particle to make mRNA when it comes into cells, and of course to make proteins and to make more genomes. Some double-stranded RNA viruses have segmented genomes. Here are the reoviruses. They have anywhere between 1 to 12 double-stranded RNA segments. This is one with 12. They can have fewer. All right, double-stranded RNA. Then moving to plus RNA, single-stranded plus RNA. We have, the plus RNA can be translated to protein directly, and then it's copied through a minus strand to make more plus strands. So the enzyme copies the plus strand, it makes a minus strand, and then the minus strand is copied again to make more plus strands. The minus strand is only made for a template. Can you view it as a template to make more plus strands? And then it's thrown away. No other function. It's not put in particles. Doesn't do anything as far as we know. And I say as far as we know. Who knows? Someday we might find it does something. So here are some single-stranded RNA viruses of plus polarity. Here are the coronaviruses with the biggest RNA genomes. Here they go from 27.6 to 41.4 kilobases of long plus strand RNA. And mRNAs typically have certain features. They have untranslated regions at either end, which are not made into protein. They may have caps at the 5' end, which is a very specific chemical structure of a base, which we will talk about later. It's important for stability in translation. They may have a poly A tail at the 3' end. Then we have the flaviviridae, 9 to 12 kb of plus-stranded RNA. Can you name a flavivirus right off the top of your head? I'm not sure. I bet you should be able to. There are a bunch of them. I'll wait for it to come up there in the chat. Meanwhile, picornaviruses, 6.7 to 9 kb, plus-stranded RNA. These are unusual. They have a protein linked to the 5' end, and we will talk about that later. Then we have togaviruses, 9 to 11 kilobases of RNA. And so what are the viruses that you might recognize here? Well, coronaviruses, of course, include poliovirus and rhinovirus. Those are important human pathogens. We are on the cusp of eradicating polio. All right, so we have some guesses for flavivirus. Yes, so we have yellow fever virus, hepatitis C virus, Zika virus, dengue virus. Absolutely. Those are all flaviviruses, not Ebola virus. In my course, I don't take away points for getting the wrong answer. How about that? Many professors do. I give you points when you get the right answer, but I don't penalize you for getting the wrong one. Then we have caliciviruses, gastroenteritis viruses. These are the viruses of whales that I showed you on day one. Norovirus, the cruise ship virus, is a calicivirus. Then our coronaviruses, SARS-CoV, MERS-CoV, SARS-CoV-2, the 3 epidemic human coronaviruses. Flaviviruses, there you go. Yellow fever, West Nile hepatitis C, Zika virus, dengue, as someone also put in there. And the toga viruses include rubella and equine encephalitis virus, among others. They all have different structures, pure protein envelope viruses. We will explore virus structure on Monday. That'll be fun. Lots of pictures. I will bring my virus models here. I have to ship them. I have to transport them down from Colombia to show you. Then another group of Ebola virus is negative stranded, which we'll get to in a moment. Here we have single-stranded plus sense RNAs with a DNA intermediate. There is one viral family, the Retroviridae, in which there are 2 human pathogens, HIV, of course, HIV 1 and 2, and human T lymphotropic virus, HTLV. This is really a remarkable virus family because of the reproduction strategy. We have a plus-stranded RNA genome in the virus particle, but when this virus enters the cell, it is not translated to protein. Instead, it is copied to DNA. First, minus DNA and then double-stranded DNA. And as you know, double-stranded DNA can then go on to be transcribed to produce mRNA, and it's also copied to produce more genomes for packaging into new virus particles. Now, the remarkable part of this strategy is that this DNA actually integrates in the host cell, which we will talk about later. And let's finally, maybe not finally, but penultimate, minus single-stranded RNA genomes. Now, minus RNA, when it comes in a cell, it can't be translated, right, because it's the wrong polarity. So you have to copy it into a plus strand, which could then either be translated or put into used to make more minus strands to put into new virus particles. But the cell cannot copy this minus RNA. So minus RNA viruses, by definition, have to have in the virus particle an RNA-dependent RNA polymerase. All right, that's very important. You can look at the polarity of the genome and predict whether there's going to be a polymerase in the particle. So, with the exception of course, retroviruses, where you would say there's no polymerase because it's a plus, but the plus needs to go to DNA, and so you need a reverse transcriptase. So these viruses all have an RNA polymerase in the particle. And these genomes can be segmented or they can be non-segmented. And here are some examples of viruses with minus strand polarity. We have the paramyxoviruses, measles and mumps virus. We have the rhabdoviruses, rabies virus, filoviridae, Ebola viruses, and Marburg viruses, very, very long filamentous particles, all negative strand RNA, influenza viruses, and arenaviruses. Now, all of these viruses have an RNA polymerase in the particle. They are negative strand viruses. This last family here, arenaviruses, is interesting for two reasons. First, that the genomes are actually ambisense. They are both minus and plus, but when they get into a cell, the plus part is not translated first. So you have to make a plus from the minus strand. So these viruses have to have an RNA polymerase. It's a bit of a complication. Don't worry about it. The other reason they're important is because Lassa virus is an arenavirus, and this book, Fever, which I highly recommend, I think it's now available on Amazon as a Kindle. It wasn't for years, so that's great. By John Fuller is the story of the discovery of Lassa virus in Nigeria in the early 60s. And I read this after I graduated college. I was a biology major. I didn't know what to do with my life. I read this book and I said, I have to be a virologist. And that was my path. And you can hear more about that. There's a podcast about my history. My podcast, Vinny, it's called. It's a TWIV. Great book. Great book. Now, when you have a segmented genome, you can do something that viruses with non-segmented genomes cannot do. And that is you can reassort. You can mix up the segments. So here's influenza virus. 8 RNA segments of negative polarity. And influenza viruses comes in different variants. So here's a red variant or an L variant, and here's a blue variant or an M variant. And if these two viruses should infect the same cell, which can happen, if you're co-infected with 2 different variants, that happens. Now the cell will begin reproducing these RNAs, and you'll have both blue and red RNAs. And then when you make new virus particles, you'll have the red virus and you'll have the blue virus, but you'll have some with a mixture. This one has 1 red RNA and 7 blue RNAs. That is called reassortment. The process that gives rise to these viruses is called reassortment. It's very high frequency, and it can give rise to high genetic diversity. And that is one reason why influenza viruses from time to time cause pandemics due to reassortment, which we will discuss when we talk about emerging viruses. And then, yes, the last category of genome here of RNA viruses is ambisense. This we've mentioned already, Lassa virus, arenavirus. It is these viruses have 3 segments, these arena and peribunya viridae. And you can see that the RNA is a hybrid. It has a little bit of plus and a little bit of minus by the color. And these viruses have to have a polymerase in the particle, an RNA polymerase, because when these RNAs come in, the first thing that happens is not translation for reasons that are not clear. You have to actually get the proteins encoded in the negative strand, and so to do that you need a polymerase to make an mRNA, which is then translated. So it's a little funny, but hey, it works. Nature has allowed it to work, I suppose. Now, so far in this course, we have looked at RNAs as lines, right? I draw them as squiggly lines, greens, different greens. But they're not. In the particle, they're all structured, and here are 2 ways of looking at that. So RNAs, even the single-stranded RNAs can base pair with each other if the bases are complementary. So if there's an A here and a U here, they can form a base pair. And so they make things called stem loops and bulges, and these can form extensively. This is a picture of a plant virus RNA genome, which has extensive base pairing throughout the RNA, and there can be long-range interactions. These red parts can base pair if they're complementary, and these blue parts can base pair, and they do. And these structures and this long-range base pairing has function, and we will talk about that from time to time. Here's an example from HIV. This is the very 5' end, and what 5' and 3' refers to we'll talk about in a couple of lectures. it refers to two different ends of the molecule, and why that is chemically so we'll show you. But this is the 5' end of the HIV-1 RNA genome, and there are lots of these stem loops here. And even the way we draw this here with the base paired stem, right? We have two lines with little ladders drawn between them. Well, those are base pairs between like an A and a U, or a G and a C. And the loop, it doesn't even look like that in the RNA. So this is the actual structure of this RNA. So the investigators purified the RNA and they solved its three-dimensional structure, and this is what it looks like. It's highly has shape, and these are the stem loops here, but they look very different from what we draw. So I just want you to remember that RNA is not just a line. It has a lot of structure that can be important. All right, I think this is our last question for today. Which statement about viral RNA genomes is correct? A, single-stranded RNA may be translated to make protein. B, double-stranded RNA can be directly translated to make protein. C, single-stranded plus RNA replication cycles do not require a minus strand intermediate. D, RNA genomes can be copied by host cell, RNA-dependent RNA polymerase, or all of the above. Which statement is correct? And meanwhile, let's go to some questions here. I wish there were a way to stop this chat from scrolling so that I could pick up where I left off. I think I sort of have it. All right, I answered that one. All right, I answered that one. What's the difference between plus RNA and plus single-stranded DNA? Well, you have the DNA has thymine and the RNA has uracil, but there are also chemical differences, right? One is ribose and the other is deoxyribose. And the sugar, there's a ribose sugar, which is part of both, and there's an extra hydroxy on one versus the other, which we'll look at when we talk about nucleic acid. So there are other chemical differences as well. Isn't recombination like sex between viruses? So it has the same function. So sex, we think sex evolved in order to provide genetic variability, in fact, to combat parasites. Because if you have non-sexual reproduction, you don't have a lot of genetic diversity, and you're susceptible to infections of various sorts. So you combine, genomes from male and female, and you get variety. And that's the same thing with recombination. Yes, you get variety in the case of viruses because they don't have sex, but they do recombine, or reassort is another way of looking at it, right? And so as you see, Sobran, do viral RNA and DNA fold? Yes, you saw, I showed you, you anticipated my my little slide there on the structure of RNA, and DNA also folds into structures as well. Does weather and cold temperatures have an effect? Yes, it's not a myth. There is experimental support, which we will get to in the second part of this series. Yeah. If RNA is fragile, how is it that the saliva test heats it up? Well, it breaks for sure. It's broken, but there's still pieces long enough to be detected by PCR, so it doesn't matter. Is an RNA-dependent polymerase known as replicase? Some people call it that. I think it's a horrible name. Because they call it replicase and transcriptase, one for replicating genomes, one for replicating, for making mRNA. Transcriptase does nothing to do with RNA viruses. It has to do with the making of mRNA from DNA. This is a thing that we have in our textbook, and many other virologists will disagree with us, but we decided not to use replicase or transcriptase. We call it RNA synthesis, RNA-dependent RNA polymerase. Do bacteria have defenses against viruses? Yeah, they have CRISPR-based defenses. That's where CRISPRs are evolved. We'll talk a little bit about that, but that's probably, we'll go in detail more in another course. Very good question. The number of people are asking about prions, and these have been answered by the mods. Prions are proteins that cause disease, and we'll have a lecture on prions. Stay with us, folks. You have a lot of good questions, and all I ask is to keep watching, and you will get your answers. And I'm glad that Priscilla is being pedantic. Hep C is the disease, and HCV is the virus. Excellent. You know that we are pedantic, right? Get the terminology right. So that doesn't confuse matters, because everything else is confusing enough. I thought cells have RNA-dependent RNA polymerase. No, they do not have an RNA-dependent RNA polymerase that is able to copy large viral genomes. They have a polymerase that can make small RNAs only. They will never copy an entire virus genome. So cells do not have RNA. That's why I said cells don't have RNA-dependent RNA polymerase that can copy long viral RNAs. Do minus-sense viruses share characteristics in real life or only similarities in how they're formed? They have some, but I think they are all enveloped, actually. So that's an interesting shared similarity, but I think it has nothing to do with the genome. What are the ribosome-looking objects in LASA? Good pickup, David. They're ribosomes. Arena's package ribosomes. You can see them in the particles. We don't know why. They're there, yeah. Good point. Yeah, well, people have answered the questions about prions. That's great. Is there evolutionary pressure to make RNA viruses increase their loops and sections? There is. They are under evolutionary selection for sure, because they're functional. And if you take them out, you debilitate reproduction in some cases. So yes, absolutely. So it's a good question, because when we look at evolution, we shouldn't just look at protein coding regions, right? We should look at regions that are able to form these structures, and not everyone pays attention to those. So the problem is that in the interface I'm using for the chat scrolls. I can scroll back, but it doesn't help me remember the last question I answered, right? I guess I could put a star next to it. Yeah, so I could star the last question I answer, and then I will know to go back to it. Excellent point. Okay. Yes, and Vanity says that I highlight the last comment. So there's a way to put a star next to it. At least I can do it, and so that's how I will do it. You see, we're both learning as we go through this course. Is all information in DNA or RNA for coding for protein only? No. So there's non-coding regions that have these stem loops, which are also important for function as well. You don't need to be important only if you make a protein. Excellent point there. Yep. Then we have COVID questions, which we're not going to answer. How can RNA produce DNA? There's an enzyme that copies RNA to DNA. It's called reverse transcriptase, and you will hear more about it. Cells don't have RDRP. They don't have a polymerase that can copy long RNAs, only short ones. And in plants, they're called RNA polymerase, but they will not copy a viral genome. That's the point. All right. Let me get rid of the question here. And let me star one so I know where I stopped. Good. Excellent. What's the story here? The answer is A, plus single-stranded RNA genomes may be translated to make viral protein. That is correct. The double-stranded RNA cannot be translated directly. I emphasized that a number of times. All plus RNA has to go through a minus strand intermediate. I made a point of saying, we throw it away. It's only used for making more plus strands. RNA genomes can be copied by host polymerase. No. Now, I made a big point of that. They cannot be copied by host RNA polymerases. There aren't any polymerases in the host cell that can do that. That's why every virus genome on the planet that we know of encodes an RNA-dependent RNA polymerase to copy its genome and to make mRNAs. and not mRNA, not all the above, for sure. All right, that does it for Socrative for today. Let's go back to slides. Now I'm going to end up talking about the genetics part of this session, genomes and genetics. This method, what is this method called? This is the plaque assay. This allowed people to do genetics with viruses. What do I mean by do genetics? I mean to isolate or identify viruses that differ slightly from each other, have different properties, and study those properties and understand how they arise. And so the plaque assay lets you pick the viruses derived from a single infection. And so you have a clonal population of viruses which presumably all have the same property. Now, in the old days, pre-1970s, if you wanted to do virus genetics, you made mutants. And the way you made mutants was to treat viruses with chemicals, mutagenic chemicals, or you could heat them. You could do all kinds of manipulations, and then you would plaque out what you got and see if you had viruses with different properties. Very arduous, but people were able to identify viral proteins involved in heat stability or resistance to certain drugs. But nowadays, we don't do that anymore. We use recombinant DNA. And before we talk about this, we need to, again, define some terminology. And here is one of my greatest pet peeves, the word mutation, which is almost uniformly misused by the press, especially the science press, but also the non-science press. A mutation is a change in DNA or RNA. It can be the addition of a nucleotide. It could be the deletion. It can be the rearrangement of it. Any of these are mutations. Mutations then may lead to amino acid changes, substitutions, additions, removal that would make a shorter protein. And that's what a mutation is. A mutation is not an amino acid change. It was originally defined as a change in the nucleic acid. And I'm not changing it. We didn't change it in our textbook. People want to change it and talk about spike mutations that, are invariants, but it's wrong. And I'm never going to win this battle, but this is where I stand. And in my class, you better restrict mutation. This summer, I taught virology, summer course at Columbia, and I had this on the final exam, and a student got it wrong. The student said a mutation can be an amino acid change. And she said, why not? And I said, well, go back to the lecture and look at this slide where I was pedantic and told you about it. Okay. In the old days, as I said, mutations were introduced by mutagenesis, ultraviolet light, chemicals, heating, any kind of selection that you want. But we don't do that anymore. Nowadays, the modern way, we have what we call infectious DNA clones of viral genomes. And we can modify DNA to introduce mutations and then hopefully recover viruses with those mutations. I call this a modern validation of Hershey Chase. Why is that? Well, Hershey Chase said the DNA genome is the genetic material, right? So if you take a DNA copy of a viral genome, whether it's a DNA virus or an RNA virus, doesn't matter, and you put it in cells and out comes viruses, that means that the genetic material is DNA, right, or RNA. So it's a validation of their experiment. You can make all kinds of mutations in DNA, and then you can make viruses by putting the DNA into cells and study them and understand the function of genes. You can make viruses into vectors for delivering vaccines, for correcting genetic diseases. We'll talk all about that in the last session of this course, therapeutic viruses. And the way it works is illustrated here. This is a bacterium. It's got its own bacterial DNA. It's pretty big. And then many bacteria carry what are known as plasmids. These are double-stranded circles of DNA. They can be relatively small, a few thousand bases or 30, 40, 50,000, but much smaller than the bacterial DNA. And we can take a viral genome. We can take a DNA genome and we could insert it into these plasmids by genetic engineering methods, recombinant DNA methods. And then we can grow the bacteria and make lots of our viral DNA. We can then change it, and then we can introduce it in a cell by this procedure called transfection. So if you have an RNA virus, you can use reverse transcriptase to make a DNA copy, and you put the DNA in your plasmid and grow it in bacteria. That's the modern way that we make mutations, and almost every experiment done with SARS-CoV-2 involves a DNA copy of the genome, which is modified to introduce mutations and then investigate their effect. So the word transfection was originally coined for bacteriophage lambda, where they found that if you extracted the DNA from lambda and put it into bacteria, that you would get virus reproduction and more lambda virus particles. So production of infectious virus after transformation of cells by viral DNA. Transformation, the introduction of DNA into cells. It's an unfortunate word because transformation also refers to the properties of cells that may lead to cancer, as you'll see later, but we're stuck with it. So making virus after putting DNA into cells. So transformation, infection, that's where the word transfection comes from. Nowadays, it's used to mean any insertion of nucleic acid, whether it's RNA or DNA, into cells, and that's fine. So you can put viral RNAs into cells, you can put viral DNAs into cells, and out comes viruses. This is how we do virology nowadays. So, for example, here's the experiment done with poliovirus, infectious poliovirus DNA. So poliovirus is a virus with an RNA genome of plus polarity. If you remove the RNA from the shell, which you can do in the lab very easily, you can transfect that RNA into cells in culture, like HeLa cells or whatever you wish, and as long as the cells are permissive, out will come infectious virus because this plus RNA is translated and it initiates an infectious cycle. What can be done is to take this RNA, make a DNA copy of it, and then put it in a bacterial plasmid so you can make more of it. You could take that DNA actually and transfect it into cells, and it will initiate an infectious cycle. Or you can synthesize RNA in vitro, transfect the RNA that you make into cells, and you will also initiate an infectious cycle. So we call this infectious poliovirus DNA. By the way, this transcription, this in vitro RNA synthesis, is using the same technology that's used to make the mRNA vaccines. We have a plasmid encoding whatever you want to make mRNA of. You put a signal in the DNA so that when you add your enzyme, it will make mRNA. And we've been doing this for years. So this is actually what I did as a postdoc with David Baltimore. I showed that if you made a DNA copy of poliovirus and put it into cells, it was infectious. The first such discovery for an animal virus, which has since, of course, been used for every other virus on the planet. One more example. For every virus, there's a slightly different way to make infectious viral DNA. This is influenza virus, where you have the challenge that there are 8 pieces of RNA in the virus particle, and you need all of them to initiate an infectious cycle. So what was done here a number of years ago, after we had done our polio experiment, they take each RNA segment and they make a DNA copy of it, and the DNA copy is put in a plasmid, but I'm showing you just the relevant parts of the plasmid as a linear diagram here. And so there are 2 promoters incorporated into this plasmid. Now, a promoter is a word we haven't encountered yet. Promoter is a signal that tells RNA polymerase to make mRNA. And so here on the left side of this diagram, there's what we call a Pol2 promoter. Pol2 is the enzyme in our cells that makes messenger RNAs. And so that promoter is actually going to make influenza virus plus strand mRNAs. And then on the other strand, we have a Pol1 promoter, which is the enzyme in the cell that makes other RNAs, not mRNAs. And that's going to make minus strand influenza virus RNAs. And so what you do is you take a plasmid encoding each of the viral RNA segments, each of the 8, 1, 2, 3, 4, 5, 6, 7, 8, and you put them in cells, 8 plasmids, Each plasmid makes mRNA and minus RNA. The mRNA goes on to make proteins. The minus RNA, of course, is going to be the genome. And out comes influenza virus. Isn't that cool? Yeah, I had it easy for polio. I just put the DNA in a plasmid and threw it in cells, and it worked. I wanted to put promoters in, but David Baltimore said, Vinny, don't bother. Just put it in cells. And boy, that's the moral of the story, is just do the simplest experiment first. It didn't work for influenza, so they had to do this. Now, what do you do with this? One of the things that was done was to recover the 1918 pandemic influenza virus, which, as you know, killed millions of people, far more than so far killed by COVID. This is the worst pandemic of the modern era, I would say. But in 1918, we didn't have influenza virus. We didn't isolate influenza virus until years later, and no one had isolated the 1918 virus. The 1933 was a different isolate. So when the system that I just described to you was developed for recovering influenza virus from DNA was developed, investigators said, let's recover the 1918 virus. So how are we going to do this? Okay, so they took, it turns out that many service people, right, died in the Army service during the pandemic, and the Army saved a lot of their lung samples. They made little tissue blocks preserved in formalin and wax, and they were stored away. And so the investigators went and got some of them, and they extracted the viral RNA, and they got some of the genome sequence from it. But they didn't get the whole 8 segments, so separately they went up to Alaska, parts of Alaska that have always been frozen, and they exhumed a person who had been buried, who had died of influenza in 1918. They did a biopsy in situ of the lung, took out a piece of tissue, and got the rest of the viral genome sequence. Got all 8 genome sequences. They put them, made DNA copies, put them in a plasmid, and they got virus out. Now, you know, this is a 1918. This is a picture from 1918. Huge spaces where sick people were cared for. There wasn't room in hospitals. We saw this with the COVID pandemic in stadiums and so forth. And so, of course, people immediately became outraged. How could you do this? You're going to start another pandemic. Well, there's a good reason to do this. We would like to know why this virus was so deadly, and if it came back, would we be ready so they could take this virus and say, do antivirals work? Do antibodies work? And so forth. And that information is valuable. It is certainly dangerous, so you work with it under very, very high containment. The highest containment that we have is BSL-4, which we will talk about later. Now, I also want to tell you that you don't have to clone the DNA of a viral genome. You can have it synthesized. And that was done in this experiment, which I really enjoy. This is a topic of a TWIV. Also, this is horsepox virus. This is virus of horses. Yes, it doesn't infect people. And it's extinct, right? But we have the genome sequence, 200,000 plus bases, which any one of you can find right now. You can go onto the data bank and get it. And so a number of investigators wanted to recover this virus for reasons, valuable reasons. They wanted to see if it would be a good vaccine vector. So they had synthesized 10 overlapping DNAs that would together make the whole 212,000 base pair genome. So each of these red bars is one of these pieces of DNA. They overlap, and together the 10 of them constitute the entire genome. That cost them $150,000, folks. So, you know, that's not cheap to do this. They took these DNAs and just put them into cells. They threw them into cells by transfection, and out came infectious virus because they all recombined with one another. And at some point, you got recombination to form the viral genome, and boom, it was transcribed, and it took off. And, of course, this was also met with horror in the press. It actually leads me into the last part of what I want to discuss today, and that's synthetic virology and biosecurity. So the ability to make viruses from DNA, and these are existing viruses, folks. You take a virus genome, you make a DNA copy, and then you put that DNA into cells. You get the virus back. We can't make a brand new virus. Nope, not happening. Too hard. Nobody knows how to do it. This is called synthetic virology. And, of course, there's the issue of biosecurity, because having infectious DNA makes some experiments possible that you couldn't do before. And so this one, bringing back horsepox, bringing back 1918 influenza virus. Here's a headline in phys.org. Scientists bring back extinct horsepox, raising important biosecurity questions. Not really. This is old tech. This could have been done years ago. Nobody wanted to spend $150,000 on it. That's all. So, yeah, you could get the smallpox sequence and do this, but no scientist worth their salt would do that. And if a non-scientist wanted to do it, well, they'd have a couple of obstacles. First of all, they would probably kill themselves in the process because they wouldn't know how to contain it. But they couldn't get the DNA synthesized, at least not here in the West, because if you sent the smallpox sequence to a company that synthesizes DNA, there are plenty of those companies around, they are bound by federal law to search it first to see what it is before they make it. They will find out it's smallpox and they won't make it for you. Nevertheless, we do have a committee here in the U.S. called the NSABB. That's what it stands for. It provides advice, guidance, and leadership about this kind of research, which we call dual-use research of concern. Dual-use research means you're going to do an experiment and it has a good goal, right? You're going to make some kind of advance that helps people. But dual use means the results could also be used inappropriately to damage people. So this committee reviews your research and says, okay, you could do this, but you have to use these containment conditions. And in my opinion, that's sufficient to make sure that these are done safely. Now, sure, a terrorist somewhere could do this on their own, and not much that we can do about that. You want to regulate bona fide scientists in Europe and the U.S. and other countries that have good goals? Doesn't make sense to me. Go after the terrorists who want to do this. And that brings us to the last point, which all of this intersects with gain-of-function research, because what I've just told you enables you to do gain-of-function research. It doesn't invent it, because we could do it before. Gain-of-function research is simply giving a new phenotype to a virus. That is all it is. And in fact, it's not limited to viruses, but because this is virology, I'm going to limit it to viruses. You could make a better yeast if you want to make better tasting beer. That would be a gain-of-function yeast. My lab made transgenic mice susceptible to polio. That was a gain-of-function for the mice. But for viruses, in the old days, we could do gain-of-function by mutating the virus or passing the virus in a different host so it had a new phenotype. Or today, you could modify infectious cloned DNA to give you a virus with a new phenotype. That's all gain-of-function. A certain prominent scientist has said gain-of-function is only the old way. And that's not right, folks. You know who I'm talking about. It can be either the old way or the new way. So here are two examples of gain-of-function research. On the left is a 1939 paper from Charles Armstrong, who was a senior surgeon in the U.S. Public Health Service. This is before the NIH. It's called Successful Transfer of the Lancing Strain of Polio from the Cotton Rat to the White Mouse. And so what Charles Armstrong did, polio virus, identified in 1906. There were no cell cultures. You had to grow it in animals. People wanted an animal model of disease. And, they could get non-human primates to get polio, but they wanted to use rodents, which are far more easier. So what Armstrong did was to passage the virus multiple times, first in the cotton rat and then in the mouse, and it became adapted. He selected viruses that were able to cause disease in mice and rats. That's a gain-of-function experiment. You gave the virus a new property. You selected variants that are able to do this. On the right is a gain-of-function experiment that's been in the news lately. Some of you may remember that several weeks ago, a Freedom of Information request was used to obtain a grant proposal from EcoHealth Alliance, which is a organization that has worked with the Wuhan Institute of Virology on coronaviruses. So this grant application was obtained via FOI request. You can find it. Just search for it. You can find the whole 700 pages. And this is a figure from it that shows gain-of-function research done with coronaviruses. This is an experiment where they have infected mice with different viruses, and the different viruses are shown here with different names. I'll explain them in a moment. And then we're looking at body weight with days after infection. So basically they're using body weight loss as a measure of pathogenicity of these viruses. So control animals that are just infected with saline, they keep their weight over the course of the experiment. And then we have 4 different coronaviruses, and all of these are from bats originally. These are SARS-CoV-1, not 2. So WIV1 is a SARS-CoV-1 related virus from bats. You can see it's in red. It causes a little bit of weight loss after day 5. Okay. The other viruses are actually WIV1 with the spike gene of 3 other bat SARS-related coronaviruses inserted into it. So recombinant DNA was used to produce 14, 16, and 31 here. Again, these are WIV1 with the spike of 3 other bat SARS-related coronaviruses. SHC14 recombinant is more pathogenic than WIV1 in terms of weight loss. So that's a gain-of-function experiment in my book. And the other viruses, I think 16 is not pathogenic at all. It's less pathogenic than WIV1. So putting the spike of 16 into 1 made it less pathogenic. It's a loss of function. And the purple one here is just as pathogenic as WIV1. So the only one that qualifies as a gain-of-function experiment is that one. The point here is that you want to know, you're doing experiments to know if these spike genes from coronavirus isolated from bats are able to cause disease in mice. Do they have pandemic potential? These are valuable outcomes. But of course, many people don't like these experiments. But the point I'm trying to make, it's all gain of function. And this kind of research but so is other kinds of research that is very different. Now, it turns out that back in 2016, Ralph Barrick at the University of North Carolina and his colleagues also did similar experiments and published them in 2016. I was asked to write a commentary. And so this is the commentary, Moving Beyond Metagenomics to Find the Next Pandemic Virus. And these are just some paragraphs that I've isolated to give you my perspective on these gain-of-function experiments. And I write, the critics of GOF experiments frequently cite apocalyptic scenarios involving release of altered viruses and subsequent catastrophic effects on humans. Such statements represent personal opinions that are simply meant to scare the public and push us towards unneeded regulation. And then I go on to point out that we've been manipulating viruses for years. We've never had an incident. I understand none of these arguments tell us what will happen in the future, but these are the data we have to calculate risk, and it appears to be low, and as shown by the Barrick Group, the benefits are considerable. A major goal of life science research is to improve human health, and if we prohibit experiments because they may have some risk, is contrary to that goal. Being overly cautious is not without its own risks, as we may not develop the advances needed to not only identify future viruses and develop methods to prevent and control them, but to develop a basic understanding of pathogenesis. So that is my position on this type of work. You look to see if the benefits outweigh the risk, and I think so far the work on coronaviruses far outweigh the risks, and then you allow them to proceed under the proper regulation. All right, so next time we are going to talk about structure of viruses. That's Monday, but let's do some questions here. Now I'm going to use my newfound method of going back to the star. Oh, where is the star? Oh, here it is. There it is. Good. It works. I can go back to see the last email I answered. A lot of talk about prions and prion diseases. We'll get to that later. Is there an advantage to retroviral replication strategy versus simpler? What's the reason? Well, the DNA is integrated into the host. So that allows it to become a permanent part of the host if the host isn't killed, of course. So I think that's kind of an evolutionary advantage. So that would be the main one over a plus-stranded RNA virus. How would you distinguish a mutation from a genetic polymorphism? So this is a really good question. So people are all different genetically, right? And who's to say that you have a mutation and I'm normal, I'm wild type, right? So that's why we call them polymorphisms, because they're just variations in the human genome sequence. There are some exceptions. There are clear areas where you have a deletion in a gene, and that's clearly a mutation that leads to loss of function, and that's a problem. But polymorphisms are where I have an A at a certain position, you have a G, someone else has a C, and they represent all forms of normal. They may have different outcomes, but there's no one wild type. I don't know if I'm making that clear, but that's my view. That's why we call them genetic polymorphisms and not mutations, because often we don't even have evidence that would say that the change is deleterious in some way, right? That's a good question. Yep. Yeah, so we call them polymorphisms, SNPs, single nucleotide polymorphisms. Yes, exactly right. And that's how I view it, that everybody's different. In fact, all the cells in your body, each one is different from the next in terms of sequence. So who's to say what's wild type, right? Are all engineered mutations done by CRISPR? No, not at all. We have many ways to engineer mutations into DNA. CRISPR is particularly useful for very specific targeting, say, of a cell, where you don't have a cloned piece of DNA, a small piece that you can readily manipulate. If you want to modify a genome, which is huge, it's not easy to do, but CRISPR gives you that ability, for sure. How can we guarantee that the current vaccine based on mRNA isn't a stable transfection? Well, mRNA doesn't integrate, right? It's a transient expression. It doesn't integrate. It would have to be copied to DNA, and there's zero evidence that you can do that. There are some papers that say it happens in cells in culture, but there's no evidence that it would happen in people. And so my view is it doesn't happen until proven otherwise. Does influenza virus count the number of segments that goes into the particle? The viruses don't count, right? But yes, it's regulated. And the mechanisms we'll talk about when we talk about packaging. Yes, so there is a mechanism for sure. How did horsepox become extinct? Not sure. I think it was control. Maybe there's a vaccine. I'm not sure if there was, but they made efforts to control it, and they were very successful at doing that. What made the 1918 more deadly? I made sure to ask what, not why. Very good. We don't actually know. Because we are limited in the kinds of experiments we can do. We can do some experiments in mice, and we can say it kills mice more than other influenza viruses, but we're not sure at all. No. mRNA may be used in transfection. RNA, that's the first thing I showed you with poliovirus. You put polio RNA into cells, it makes virus, and then the RNA goes away. It's not incorporated into the genome. Yes, the PDFs are all available on the website that I showed you earlier. Thank you, John, for your contribution. Really appreciate it. It's only now that I finally understand what you keep repeating on Twitter, that changes in spike is not mutation. Well, I'm sorry we didn't make it clear, and I'm glad you understand it now. Was I the first to transfect polio? I was the first to make DNA and transfect it and get virus, yes. And you could say that's why I got a job at Columbia, and that's why I'm here today teaching you. It's all a continuum, and I'm glad it worked out that way. All right. Importantly, the type of GOF that I showed does not qualify. That was prohibited by federal funding. For sure, yeah. Otherwise, they couldn't have done it. Yep. Thank you, James, for your contribution. Really appreciate it. All right, that does it for the questions. That does it for this session of Virology Live. We'll see you next time, Monday. In the meantime, be safe and do the quiz online tomorrow. Bye-bye.